Thank you, everyone. I'm Kiarash. I'm a PhD student at the LMB. Today, I'm going to be talking about automated model building and protein identification in cryo-electron microscopy maps. So, what is cryo-electron microscopy? Basically, for a lot of science use cases, we would like to know the structure, the atomic structure of a protein. And one very popular way to solve this problem nowadays is cryo-electron microscopy. And that includes getting the protein that you would like to solve the structure of. Usually, you express it in some bacterial system, you purify it, then you freeze it in very thin ice. And then once it's frozen in very thin ice, you basically shoot electrons at it in an electron microscope. And the problem here is that you have a 3D protein. When you do this process, what you get outside is basically 2D projections of your protein. And you would have to then solve the 3D structure again using some algorithms in order to do that. At the very end, you get a volumetric representation of your protein um, that looks like there on the left. And, that is you might get within 3 angstrom, 2 angstrom resolution, very, very high resolution. But that is not exactly an atomic model of your protein. It's just basically a blob that is the result of your atomic model being, you know, projected onto uh, the map. So how do you actually get the atomic model from that volume is you get your biologist and you have them stare at the volume and also know things about your protein and then basically place atom by atom the full atomic model. And this can take months of manual effort and require very, very specialized skills. Usually, it's the biologist who's working on the protein to do this. So, with the advancements in machine learning and so on, we thought it would be a great idea for a PhD project for me to try to automate this process. And this took quite a lot of work, and today I only have a few minutes. So I will just quickly explain how that happens and then focus a little bit on the bulk of the work. So it's a machine learning program that takes in your initial volume and first places centers of where your protein residues are, and then takes those and tries to build a full atomic model out of it. And that looks, and then there's a post-processing step at the end. And uh, how the main issue of the problem is that you actually have three different kinds of information that you want to integrate together. It's not like a usual machine learning problem where, let's say, you have text that comes in and you want to output text, that's ChatGPT, etc. Or you have videos that come in and you output videos or classification. This is quite an interesting problem because you have your initial cryo-EM map, which is a 3D voxel field. It's basically a cube of little floating point numbers for each voxel. You have text representation of what the protein sequences are in your protein. And then you have the graph representation of basically the initial points of your protein. And that's also predicted in an earlier stage of the model. And you have to get all these things to talk to each other and output an atomic model that looks very different from each of the inputs. And the way we do that in ModelAngelo, which is the program I wrote to do this, is we have a three basically module graph neural network. This is a neural network that operates on a graph because the proteins are represented as a graph. And you basically have different modules that are specialized for each of their inputs differently. They extract information from that input and then put it into a shared library of information. So you have a module that takes a look at the map and extracts information from that, puts it in a shared library. Then that shared library gets passed on, and you have a language model, very similar to ChatGPT, but for proteins, that looks at your protein sequences and extracts information there, puts it in a shared library. And lastly, you have your initial graph. You have a graph neural network, it takes information, puts it in a shared library. And then as this shared library gets richer and richer, you're getting more and more information about what your actual full atomic model looks like. And at the end, you can then predict the full atomic model. So the graph neural network is quite complicated and a lot of things go on into it. The full details are in the paper, but it looks something like this, where basically information is flowing throughout all parts of it to extract information from each different piece of input and then getting recycled back in. And so you might be wondering, how well does this work? So the whole point is, as the resolution of the map gets better and better, we are able to actually approach 100% completeness of the atomic model. That means that something that might have taken months for scientists to do manually, we can now do in an hour or two, as well as the scientists would have done. And at parts where the resolution is lower, uh, ModelAngelo is quite conservative, so it will refuse to build, which is also actually a lot of biologists will also refuse to build unless they have other information um, available. We compared it to some other more traditional ways of automating this process, which were based on X-ray crystallography data. Basically, we are able to, across the different resolution ranges, beat all other approaches, and we're able to automatically build things much earlier, as in much worse resolution than other programs. And here is a full ribosome that we've been able to automatically build. This would have taken months just a few years ago to actually go through, because it would have 10,000 different protein residues. That's something around 120,000 different atoms that biologists would have to do. And we can do this fully automatically. In the black outline, you might see that that's what the actual deposition is. That's what the biologist has built, and the colored portion is what we built automatically. So it's very similar, as you can see. And so this was the main part of my project, to just automate what people had been spending months to do. But then we realized that we can actually do better than that, and we can solve a big problem we have now in cryo-electron microscopy, which is it's fine if you have a protein in mind, you express it in bacteria, you purify it, and then you solve the structure. But nowadays, what people really do is they take proteins, purify them from cells, so endogenous sources, and for big complexes, that isn't just your specific protein of interest. It's a complex of different things and can include many different proteins that no one had associated to that complex before. And now all you have is some volume with shapes, and you don't actually know what everything that is included in that volume is. So the task becomes to identify these proteins from the cryo-EM map alone, and we use the same kind of post-processing we do in the program that I didn't really talk about today to actually extract this information. In short, we take the volume, we use ModelAngelo to, without giving it sequence information, to predict the amino acids as probabilities. Then we use these probabilities to build a hidden Markov model, which we then use to search against proteomes, like all proteins that are in the human sequence, etc. So here's a very cool use case. This is still the largest cryo-EM map solved. Um, this is a phycobilosome. It's kind of like a radar that uh, concentrates light for energy. And this specific phycobilosome uh, has more than 1.2 million atoms involved. This took months of a few PhD students to actually build the full atomic model. We were able to do it automatically in um, just a few hours. But that's not the main part, because when they actually published this paper, they had 6 protein chains that they were still not able to identify, even after having spent months doing it, because they had identified the other protein chains. We were able to very kind of easily resolve all of them to a high confidence. We told the authors, and they agreed with the identifications as well. We did the same thing in axonemes. These are basically these massive structures in different cells. Again, this is work of months of postdocs and PhD students actually identify each of these proteins, because most of them were not identified before as having to be associated with these axonemes. They still had 6 unassigned proteins that we were able to assign confidently. And these are the other 2. As you can see, some of these proteins are very unstructured. It's very difficult to actually identify that protein just from the structure alone because there really is no structure. So you actually have to have the information from the density available to then get the sequence and then search that. And so, this program has now been available for a few months, and a lot of people have used it. And there were some very cool applications, including this one, where they were able to find dozens and dozens of proteins in sperm microtubules with ModelAngelo. And so I'd like to conclude now that basically we were able to get to human parity in building proteins when the sequence is known and the resolution is high, up to 3.5 angstrom. But now we're much better than humans at identifying new proteins from the map alone when the resolution is high, again, up to 3.5 angstrom. As the resolution goes down, it's much harder to build. I'd like to thank my supervisor, Shores, co-author Dari, our collaborators, Lucas Shao, Alec Brown, Rui Zhang, and scientific computing at the LMB. Thank you very much.
Yeah, that was a brilliant talk. I guess there are going to be some questions. Can I ask, does AlphaFold have any bearing on this?
Yes, so AlphaFold helps you predict protein structures from the sequence. It performs less well when you have multiple proteins, so in massive multimeric complexes like the phycobilisome I showed, and also if you don't have sequences available, which is a lot of the problem we were trying to solve, then you cannot use AlphaFold. So AlphaFold definitely comes in when your resolution is lower, but you know what's in the multimeric document.
Interesting. Yeah.
Thank you. It was a really interesting talk. Can I ask, how does it scale with the size of the protein? So if you showed this really big protein complex, 1.2 million atoms, and that took a few hours, presumably if it's much smaller protein, it would take a lot less time?
Yes. So it scales linearly. We tried very hard to make sure it scales linearly instead of cubically or so on. But basically, we treat each neighborhood of atoms kind of independently, and so you can parallelize over those and then slowly pull the information in. So, but it still needs to process all the atoms, so it scales linearly.
If I could ask a follow-up question, more sort of like a big picture style question. I know cryo-EM has come a long way in the last 10 years. How much would it cost from a user perspective to purify the protein, express it in bacteria first, purify it, then go to a cryo-EM machine, take the data, then analyze it? What would the cost be from going from the step one of purifying the protein to then getting the structure? Do you have a ballpark of how much that would actually cost?
So one thing I know, basically it depends. One part I do know is that every kind of a session, like day-long session on the very expensive microscopes on its own is something like 3,000 pounds. At least that's what it costs the institute. The protein purification step could be quite cheap if it's a very easy protein to purify. Could be a few £100s on its own, and then that could actually just be it. So around like maybe 5,000 pounds for very well behaved, easy protein to deal with. But most of the time now you're not really dealing with those because those are very alpha predictable. So then as it gets much harder, then you also have many rounds of optimization required, and you go back and forth to the experiment, and then it could quickly scale up.
Thank you.
But whereas PhD students solving structures, that's free, is it?
Yeah, that's very free.
Right.
Hi, that's really great. What you seem to have shown is a proof of method. You can prove that it works. But what real world problems do you anticipate being able to solve, or have you not got that far yet?
No, so it solves quite a few real world problems now. For example, there's a group in I think San Diego that they do very cool stuff where they have nanobody libraries against a specific target. that they can't really identify easily which ones are bound or not. So what they do is they just put the nanobodies against their target, solve the structure of their target with the nanobodies bound, and then they use ModelAngelo to actually kind of sequence it back from the map. They get a small library, and then they can actually go ahead and identify those specifically. And there's many other projects that have now used. Yeah.
Last question.
The pictures of the big proteins, I can't remember the name, the fellow.
Phycobilisome.
Yeah.
Yes.
So you identified a handful of proteins that the humans hadn't been able, the biologists hadn't been able to identify, but they were known proteins, weren't they? They were catalogued already.
Yeah, so these proteins were available in the genome of the organism, but they did not know what the function of those proteins were, or if they were actually even present in their map.
So do you need to know every protein that might be present?
So
What would how would the AI react to an unknown protein?
yeah, so we also did this where we had a sample from like a infant's liver, and they solved some weird protein that looked like it was virus associated, but was definitely not available in the human genome. So you can then go and search against the whole proteome of proteins we know, and then we were able to identify it as part of some bacterial infection. So you can do that, and even if you don't find the exact protein, you can find very related proteins and kind of work backwards from that.
Thank you. That was a beautiful talk. You managed to explain a huge amount in a very,
Thank you.
very effective way. Thank you so much.
Thank you very much.